All right, so thanks everyone for joining us today. I hope you all had a great week. Today we have the pleasure to host Smita, who is an associate professor of computer science and genetics at Yale, and she will be visiting the CRM at UDM for the semester. So today she'll share with us some thoughts on inferring and characterizing cellular and neural dynamics with deep geometric and topological models. Before handing the mic over to you, Smita, let me remind you all of some house rules. As usual, clarification questions will be allowed to be asked during the talk. For people online, you just, in person first, you just have to raise your hand and wait for me to give you the mic so that people online and in the recording can hear you. And for people online, you just have to type the word question, or I have a question in the chat. And when the time is right, I'll let you directly ask it. There will also be some additional time at the end of the talk for people to ask further questions, make some comments, or have a more informal discussion. That's pretty much it for me. So whenever you're ready, Smita, floor is yours.
Okay. So I just do want to say I can't really see the chat.
Oh, no, no worries. I'll be monitoring.
Okay. All right. Thanks, everyone, for coming. And I'm excited to talk to you guys and actually also to hear your thoughts on some of what I'm going to be talking about. So I've been working with biological and cellular data and trying to design deep learning and machine learning models for accelerating discovery in the biomedical sciences for a while. And one of the tasks that has come up is actually that a lot of the data that we measure in the biomedical sciences is sort of static snapshots, literally like a static picture of a dynamic entity, and it turns out the dynamics are actually really important. You can imagine there's some dynamic process that converts normal cells to disease cells, or a dynamic process that has turned you from a single cell into who you are today. And so this is a really important task. And in a way, it also gives us, for us methods, people who are more interested in the methods, a chance to test out our deep learning and machine learning systems to see if they're really up to this task, which I would argue is in a lot of ways more challenging than some of the more typical tasks we may be sort of benchmarking on. So that's what I'll be talking about. So just in the last 10 to 15 years, there's actually been a revolution in biology in terms of the engineering of tools that can measure biological data. So the most famous one that people can think of is single-cell RNA sequencing, for example. So single-cell RNA sequencing basically made biological data big in the sense that we have a lot of observations, thousands of cells, and we're measuring thousands of features on these. So it suddenly let in machine learning people like me to actually enter the field and see if we can actually use machine learning to help discover insights. And you also have to remember that historically in biology, people would do small focused experiments that were designed to validate very specific hypotheses that the person already had. But the paradigm now is very different. You often have biologists generating high-dimensional, high-throughput data, just actually even hoping to discover what the hypotheses are. So this is what gives us a chance to use machine learning for everything from hypothesis, generation to making predictive analyses. And what I'm saying is not just true for cellular data, which I'll talk about for a large part of this talk, but also things like neural activity data. That is also a high-dimensional, high-throughput measurement. So here you can think of each time point as a data point. And you actually make a movie of a person over hundreds and thousands of frames. And at each point in time, you see the activation of some unit in your brain as far as how active is it. And different technologies measure that different ways. fMRI measures what's called the bold signal. And similarly, you have healthcare data and even molecular data, which I'll be talking about less, but a lot of these kinds of unsupervised methods that I'll be talking about for high dimensional, high throughput data also could apply there. So I was talking to you a little bit about how data in biology can really challenge machine learning. Some of the reasons are because the data is very, very noisy. It's so when you actually get a data point, it's largely corrupted. So there is a heavy emphasis on denoising the data, and I would say the order of magnitude of that is much higher in biomedical data than other data types. It's very, very high dimensional. I measured the single cell RNA-seq measurement itself has about 20,000 gene transcripts that it's measuring, but that's not, most of the cell or anything. These days, you can also get something called single cell ATAC-seq that can measure hundreds of thousands of chromatin accessibility locations. So the dimensionality is very high. And the data is often unlabeled because actually even biologists don't know what we're looking at. So if I take 100,000 cells from your blood, actually biologists might not even understand all the states of those cells. So that's where the burden for unsupervised discovery, dimensionality reduction, denoising, these kinds of things come up. And so some challenges directly for deep learning are, of course, interpretability. So can we make lower dimensional, more understandable, interpretable embeddings of the data? Because eventually we want to understand what's going on in the data. Can we make predictions that actually follow biophysical, biochemical constraints? How do we put these constraints in our systems? As you know, neural networks, it's hard to explicitly constrain them. So you'd have to kind of bake in the constraints in the form of inductive biases, either into the architecture or some of the operations or via penalties or regularizations. And oftentimes, today I'll be showing you a lot of learning on graphs. And it's important for our networks to actually be expressive enough to explain the complex phenomenon that happens. And we see this as particularly the case with a lot of graph neural networks. They don't seem expressive enough to express what we want them to in the bio. So some particular tasks of biology were already discussed last 2 slides. Some tasks that you might want a machine learning system to do is, first off, remove the noise and give us back the signal and the data. So this was a key problem that I was working on for nearly the first 2-3 years of my faculty position. How do we just remove noise from the data and restore the data? And you can imagine deep learning has something to do here. We have denoising autoencoders and things like that. Turns out that just that is not enough. There's a need to distill the state space into very low dimensions to be able to intuitively understand it. So even visualization takes on huge importance in biology. If you can reduce your data drastically to 2 dimensions, for example, you might be able to see what's going on in the data. If there's groupings, there might be progressions, what are the different granularities? There's another challenge in terms of multimodal, multi-sample integration. There's different modalities of data you can measure on the same entity. And then there's a question of integrating or unifying them. I'll be talking a little less about this, but the main thing I'll be focusing on is this task of learning dynamics from static snapshot data. So the cellular measurements, for example, which is what I'll talk about, when you measure those cells, they all unfortunately die because, among other things, you have to break open the cell to get all the transcripts. So that means that you haven't been able to follow a cell continuously in time. There are modalities that can follow a cell continuously in time, some kind of imaging modalities. I'll show them to you towards the end of the talk, but they usually measure 1 or 2 signals in the cell, not 20,000. So when you have this more high dimensional picture, typically, or I can't even think of a case where it's non-destructive, the higher the dimensionality is. And so in that case, the best form of dynamics that we can measure from the raw data is coarse-grained handful of time snapshots. So typically I have biological collaborators that come to me, and they typically have 4 or 5 time points that they've measured either days apart, weeks apart, or maybe even months apart. And Alex is nodding because I think I've given him many of these data sets and told him to make sense of it. So these can be spaced out far apart. But for example, here you see there's something going on, right? The data distribution is changing over time, and it used to look like the cells, if each dot is a cell, let's say here, were more similar. They're all forming one cluster in the beginning, and then they start forming two clusters, so they kind of diverged, right? So some things you might want to know is, exactly when did this divergence happen, maybe. So, you might want to interpolate what this distribution looked like at some point that you didn't measure. And then also you might want to say, okay, what is it? What was it about cells that ended up in the top cluster versus the bottom cluster? Like, could we have predicted that some cell would end up in the top cluster? And if so, where in this original cluster did it come from? So this, I would call it, the way I just asked it, a cell of origin question. And this turns out to be hugely important in biology. For example, we want to know where our metastatic cancer cells come from, and we want to know which cells, give rise to, for example, our heart, because that's how we develop heart defects and things like that. So you want to almost trace back and understand this kind of cellular trajectory. Once we have the dynamics, there's more tasks to be done. And this I'll particularly show in neuroscience. Actually, in neuroscience data, our time resolution tends to be a little bit better. Sometimes it's 1 hertz, sometimes it's better. But still, we see there's some kind of high-dimensional massive dynamics going on, which I've visualized with a method that I might briefly explain called T-Fate from my lab. But you see there's different types of dynamics going on in different people's brains. And there's a question of how do you quantify and characterize these differences in dynamics, because we don't know exactly what it means, and we're trying to understand what it kind of means. So, so those are some of the challenges and the tasks that I'm really going to be talking about today. But now I'll talk to you a little bit about why we need to use specific priors or concepts from math to help our deep learning and machine learning methods. And one of the concepts that's helped me, since I started in biology, basically, is what's called the manifold assumption. This manifold assumption is thought to hold broadly in scientific data, that this data, even though it's very high dimensional, lies in a lower dimensional latent space. And what's more, that this latent space is locally Euclidean, so you can find sort of near neighbors of cells using a simple distance like Euclidean distance. But we don't have access to this actual manifold. Well, we have access to our samples that are somewhere on the manifold, and so we have to infer, roughly speaking, the shape of the manifold from the data. And as I told you, it's not strictly lying on the manifold. It's maybe perturbed from the manifold with a little bit of noise. But assuming the noise is going in all directions, you can kind of re-infer this grid-like shape that I've shown you to some level of granularity. So you might be wondering about this, intrinsic lower dimensionality that I mentioned. Oh, it's like, oh, that's really convenient. Why is that the case? And really, that really helps us learn. And the main reason is because genes are not acting independently, like in the left feature, in the left graph. The gene feature space are not sort of independent samples or IID samples or something like that. Each gene is part of a vast network called the gene regulatory network, and each gene can induce the expression of another gene, can induce the expression of another gene, and they're all connected and mutually informative. And that's why the state space is restricted to much lower dimensions. And you can see you can learn this shape if you have a lot of samples along it. And that's one thing we're starting to get more and more and more samples all the time. So as I mentioned, so you don't have direct access to this manifold, so we have to learn it. And one of the ways we learn it is via graphs. So the basic way is to take this point cloud data and create some kind of affinity or adjacency graph from it. And some typical steps, but not the only steps that'll do this, are to take this matrix, which you can think of as a cell by gene matrix, and compute distances. And as I told you, the manifold assumption allows us to pick up simple distances like a Euclidean distance. And then you get a distance matrix. And you see the distance matrix is symmetric, and then now it's a cell-cell matrix instead of a cell by gene matrix. But this distance matrix doesn't really clarify the manifold because it's also using Euclidean distance on non-local connections. And as you saw in the manifold, non-local paths would be along the manifold and not found by Euclidean distances. And so one of the steps we perform in order to get an affinity graph that can be a rough model of the manifold is passing it through a kernel like this. This kernel highlights the small distance connections and de-emphasizes the large distance connections. And in another guise, you can think of it as a kin-n matrix. So if this was a k nearest neighbors or something with a strict cutoff, then you have a kernel that's kind of a box shape for each cell, and then you are only highlighting the near neighbors. And you can see this does turn the data into a weighted graph that does follow the shape of the manifold. So this is a really important step. And so a lot of the networks that I'll be showing you either directly have graphs embedded in them or they have graphs dimensionality reduced into some coordinates. And so one other operation that we've found really, really useful is what's called a diffusion operator. It's computed directly from this affinity matrix. You take this affinity matrix that has some affinity value between two cells, I and j, and you row normalize it by using the row sum in the denominator. And so this creates a Markov matrix, which can also be thought of as a transition matrix. And the transition is describing is a random walk on this graph. It's saying if I'm on one data point, which is one cell, what's the probability I jump to other cells? And then you can raise it to some power to compute probabilities of t-step random walks. And it turns out that if you're computing the probability of walking from this cell to this cell, because it's restricted to jumps on the data, it will not jump across the manifold like the Euclidean distances we're doing, and you get a very faithful, more global distance between points along the manifold. So the diffusion operator turns out to be very important. And again, if you had an adjacency matrix, you just compute it by normalizing. And in a way, what this diffusion operator could also say, instead of thinking about it as a random walk, is if you started with a unit of heat here, how would that heat spread? That is another way of thinking about the diffusion operator, and that's why it's called diffusion, sometimes called random walk matrix. And it's been proven by many people over the years, and a lot of these papers are 20, 30 years old, that diffusion on a manifold can recover manifold distances, and that this discretization of a heat diffusion operator, as we have with the diffusion operator, gives us manifold distances on a manifold graph. And so that's another reason we tend to utilize it. So we often use what we call diffusion geometry. So this kind of idea that we can get distances between points on a manifold using this kind of diffusion operator, together with some topological quantities along with deep learning to make systems that can make sense in biology. And this is kind of a combination that we've used many, many times in different papers. But today I'll talk to you about how we're using it specifically to understand dynamics. So the work that I'm first going to talk to you about is a paper called MioFlow, which some people in this room were also involved in developing. And MioFlow was published in a version in NeurIPS 2022, but we're continuing to work on it, so hopefully we'll come up with a journal version soon. The idea of MioFlow is to take that manifold graph that we created from the diffusion operator and create an embedding space. And this is done by using an autoencoder that's penalized to keep manifold distances. And then in this space, to create a dynamic model in the form of a neural ODE that infers the dynamics. And the way it infers the dynamics is using the concept of optimal transport. So it takes data that you've measured at a particular time point. It computes trajectories for every point using the ODE, and then it advances those points to a subsequent time point. And there you can match using the concept of optimal transport. And it turns out that you can also get this as a system to do dynamic optimal transport. I'll tell you in a second. So again, here's a data example where we have 5 time points. 1, 2, 3, 4, 5. Okay, those are the 5 static time points, and we want to make continuous trajectories from. So this is a very popular problem, as I mentioned, and it's a RNA velocity. RNA velocity tries to look within the transcripts to see which are new and old transcripts. Go ahead. Oh, those slides aren't following? Oh, like the
slides you're reading from your.
Yeah, it's not following. Oh yeah, it has not followed.
It has followed, yeah.
Wait, do you think I'm disconnected?
Can you just close the window?
Which window?
The PowerPoint window and go back to the meeting. No, not good. That's okay. Can you share it again?
But how did that? Didn't I just close it?
Stop presenting and present the entire screen again.
Okay, so you want me to present the whole screen?
Yeah. And go back to PowerPoint.
Now reopen it. Okay.
Okay.
Now do this mode. Is it okay?
Yes. Okay, perfect.
Wait, so when did it stop following?
Well,
Just
the
when you-
milestone slide,
Uh-huh.
and then you're talking about when you said 5 different time points, and you were
Again.
following I think you're showing with
Yeah.
your cursor.
Oh, that's when it
Whatever
stopped.
slide is maybe after that, we didn't see. Yeah, so this we didn't see.
Okay.
The
And the
milestone.
next one. This one we saw it. It's following, I think.
Well, not on screen.
No, this one.
And then this one. Not.
Okay. This is the slide I was on anyway. That's weird that it suddenly stopped, but
Yes,
thanks for
thanks.
alerting me this. I hope let me know if that happens again, I guess. Okay, so now I'm on a different slide. Okay. This is crazy. Okay, let's see if it'll
Yeah, that might
see.
be
It's like one slide behind where I am.
I think that's presenter mode. So this is showing you either a slide ahead or behind to get you prepared.
Okay, now it's even with this, so we'll
Okay.
see. Okay, hopefully it stays. So I was just showing other advances in this area. So this is RNA velocity gives you kind of an estimation of where the cell will measurements of which transcripts are new within the cell and which transcripts are old. But you can imagine that because the cellular measurements are already so noisy that this is an even noisier calculation. So a couple of disadvantages are it tends to give sort of conflicting information for nearby cells, but also it has no ability to bridge across those gaps if you have spaced out measurements. And so it doesn't really give you the whole trajectory of a cell. More relevant piece of work is this work that's called Waddington OT. So Waddington OT maybe was one of the first papers that started using the concept of optimal transport, but they used it more as a matching. So you get kind of an idea of which cell is like this cell at the next time point, so kind of matching. But what we actually wanted was, the full cellular trajectory. So we're sort of a plausible cellular trajectory so that we can sample the trajectory anywhere we want and give out kind of continuous time dynamics from it. So that's our thought. And we started it with TrajectoryNet, which was a work that Alex, Guy and others were involved with in 2020, which is kind of an earlier iteration of MioFlow. So those are some other works. So as I mentioned, MioFlow first uses an autoencoder, but not just with the reconstruction loss. As I told you, that doesn't seem powerful enough to, preserve biological manifolds, which tend to be very nonlinear. So actually, we penalize this autoencoder to keep the data diffusion geometry. And in the MioFlow paper, we use multiple scales of the diffusion operator in the sense that we use different scales of diffusion. So we're basically taking the diffusion operator instead of to 1T to several dyadic scales. And this preserves a multi-scale diffusion distance. In a previous work, also by Alex, in diffusion EMD, we actually showed that if you have a multi-scale diffusion-based density estimation of any signal on a graph, that this is very related to the Earth mover's distance just by taking in a one norm of the difference between these densities. So it's very easy to compute also Earth mover's distance in this particular space. So that's some of the reasons why we use this space. And we can also show that in this basically optimal transport distance, the ground distance is a manifold distance. So within this space that has some interesting properties, it's preserving diffusion geometry, optimal transport is easy to compute. We solve a neural ODE. So the ODE is just a neural network that produces an instantaneous time derivative, and then the ODE solver integrates it out. And there's optimizations to avoid backpropagation through the ODE solver, which can be computationally helpful. And then what you can get is the trajectory at any time point that follows. So as I mentioned, the ODE solver computes trajectories for a whole cloud of cells, and it pushes the whole population forward. And the penalty is not really to come up with this trajectory, but just to match the marginals. But in matching the marginals efficiently, it produces the trajectory. But of course, we want our trajectories to be as efficient and as smooth as possible, and not to just match the marginals. So we constrain the magnitude of the derivative that comes out of the ODE, and that's what gives us these very smooth, energy efficient, plausible trajectories. And we're able to show that if we penalize the neural network to doing this, then, and we have some way of matching the marginals as well, that we have what's called a dynamic optimal transport rather than the regular static optimal transport. The idea of the dynamic optimal transport is that not only are you displacing these points, but you're considering the length of the path to get them there, so you don't have these wasteful kind of slides down the S here, and instead you're going directly to where it belongs. So this is an example that shows that. And the more you hike up your energy penalty, is this penalty right here, you get it to be more and more like dynamic optimal transport. But you might be thinking, oh, well, it's nice and good to transport cells from one state to another, but that's not really what cells do. They also die and they also proliferate. So it's not purely a transport problem. So how can we account for this? So we do a couple of things. First, we've switched from the standard OT to match the marginals to unbalanced optimal transport, which can allow some of the mass to not be transported. And the second thing is we actually are now learning a time-varying growth rate for each cell. This is not part of the original paper, but will be part of the journal version. So here's an example of MioFlow doing this. So you see that if you have this kind of branching data where the branches have different amounts of density, if you're restricting the matching to all the cells, you'll see some spurious trajectories that emerge. And now you see that if you learn the growth death rate, so this is a rate that's either over 1 or under 1, and you learn it at each instance in time, you can get continuously model changes in kind of the mass of the cellular population. And here's changes in sort of predicted masses of the cells that you're seeing. Lately, we've also experimented with an SDE, which can give you somewhat squigglier paths, but it can also do this. And then we've modeled dying. So these cell populations are slowly being kind of eliminated. So populations are eliminated or more down from here and growing. So this turns out to be really important. And so now we're not just computing one time instantaneous derivative, but 2 of them. So this is actually a PDE that's being, and it turns out that that analysis is very important. In a recent comparison, we've sort of compared. No, it didn't change again. Oh, there we go. Okay, in a recent comparison, we've compared MIA-flow to different things, sort of quantitatively. Quantitatively speaking, it's possible to compare against other flow methods. Qualitatively, you can compare against things like RNA velocity and even pseudotime, because RNA velocity just gives you arrows, it doesn't give the full trajectories. And diffusion true pseudotime just gives you one axis of progression. So over that, the flow-based models can give you actual trajectories, but you see that if they're not completely regularized correctly, they can cut across space, they can give you squiggly, energy inefficient trajectories, or a variety of other problems. And the way we make a comparison like this is a little bit tedious. First, you have to use a simulator for cells. So we used a Sergio simulator. We had the ground truth network and ground truth trajectories, and we generated sampled cells from them. We try to compare the ground truth trajectories gene by gene with the original ones, either using optimal transport or error in the latent or ambient space. And you see that either the error or the earth mover's distance is smaller with MIA-flow. So now that you have this and we've sort of validated it, you can actually transport your populations, and you get, at the end of the day, single cell trajectories that go from the beginning to the end time point. So we've been wanting to apply this to study disease, particularly cancer. So one kind of cancer that we're studying together with our experimental collaborator is breast cancer. And it turns out breast cancer has a process called EMT because it's an epithelial cancer. So your breast cells are epithelial cells, and these cells have to quite transform themselves in order to seed a metastatic tumor. First, epithelial cells, like your skin cells, are on a grid. They have to become unstuck. They have to gain the ability to swim, and they no more epithelial type. Now, processes. We're studying these processes over, again, sort of a 4-day time course, spaced out. And you see there are single cells seated here. These are cell lines of a breast cancer cell line, and they start growing these kind of small tumor-like formations called mammosphere, and they become bigger and bigger over time. And these were all sequenced using single-cell RNA sequencing, giving a total of some 18,000 cells. And when you draw them with our dimensionality reduction method FATE, they sort of look like this. Yeah. Oh, yeah. No. I don't understand this. Did it change? It changed online though? Okay, so this is the animation with the time, but I'm not gonna maybe it'll force me. Okay, so anyway, so we ran the trajectories on that data set, and one of the important analyses that comes out of it is a cell of origin analysis. So we can show that in the original time point, not all the cells are transported, and some are transported, and they have a very specific characterization in terms of genes. So basically, if you trace back the cells that make it to the last time point, it seems that they come from an area of the cellular manifold that is characterized by these genes. And this we're actually able to validate. So in the preprint, which is online, you can take a look. We seeded cells that were high in EpCAM, CAB1, and CD44, and it turns out that even partially the signature enhances the ability for cells to transition. So that's one kind of analysis. The next kind of analysis is to actually analyze the trajectories. Did it change online again? Yeah, so we have two different types of trajectories, and we want to understand what's different about these two kinds of trajectories. And so it turns out that the left trajectory is that reversion back to the epithelial state, which can form the tumor. The right trajectory, those cells are going to die, so they're harmless. So what we do is we decode back out of the autoencoder space, and we study the gene trends along time. And we can see that the gene dynamics are actually different between the two kinds of fates, the tumor forming and the non-tumor forming. And we can begin to kind of form ideas that are time-based on what gene might be starting off this event and what gene it might trigger and what gene that might trigger. So trying to understand the ordering of events that happens. In order to do that, we actually take the time trends of each of these genes and we cluster them. So we get kind of a dynamic picture of the cell clusters. I don't know if that's coming up. Yeah, I don't know either. I think it's this computer. And so the final thing that we do, should I stop and restart or?
Yeah, you just stop sharing.
Yeah.
Don't close the PowerPoint. Yes. No, let's try to stop sharing. And it's the entire screen. And we can go back to PowerPoint. Sorry about that.
Okay, so anyway, we applied a simple statistic called Granger causality initially to get an understanding of which time lag genes are predictive of one another. And we developed a network from our genes that consists of 4 gene clusters, and these gene clusters are activated in order. So that's the understanding of the event ordering. And these arrows that you see are a measure of the Granger causality between these genes. So once we have this, we sort of have a network that might be driving this kind of metastatic. In that network, we picked out a subnetwork, and this subnetwork is interesting because at the bottom of the subnetwork are very well-known genes that control EMT, but at the top are cell genes. And it turns out that specifically these 2 genes are, the top gene has hardly ever been reported, and the second gene that's up from here, which is an androgen, so it's related to the androgen receptor gene, is under clinical trial right now. So androgen receptor inhibition, and they're showing that it improves overall survival. Is doing it again. We've also tried inhibiting Ezra, which is the top gene in the cascade, and we're able to show that if we inhibit Ezra, it drives MET, blocks EMT, so it should be able to be used as a modulator. So these are some kind of novel hypotheses that we can come up with by modeling the continuous trajectory, which then gave rise to this network learning paradigm. So the next thing that we wanted to do is we realized that using causality was not really how we wanted to learn these gene-gene relationships. Because just like the cellular trajectories, these gene networks are also dynamic. And so one of the things that happens when a cell changes from an epithelial to a mesenchymal cell is the underlying gene network itself sort of changes. And we wanted to see if we could also learn the gene network sort of using the proxy loss of trajectory prediction. So kind of, in a way, make the neural ODE also give us this gene network. So in order for it to do that, we actually we created a different ODE network, which is a graph ODE, and we put the graph of the gene regulation right into the network. And this was also helped by collaborators Frederick, who's back there. I hope I spelled his name right correctly. And then Guy. Frederick knows I have a long history of trouble spelling his name for some reason. So then in this Routini architecture, the task again is to predict the gene trends. But now the gene trends are continuous because we are able to infer them into continuous time. So this gives us a lot of ways of penalizing this network. And the way it has to do it is each gene has to pay attention to its neighboring genes, which are the genes that it's interacting with or being activated by to predict the next one. But initially, we have some random guess. We can even use the Granger causality as a guess. But the network updates itself using a special attention mechanism that we designed. So the attention mechanism that we designed, we call it the space-time attention. So it's a type of attention that's over time and over the neighbors. And so what that allows is for there to be time lag between a neighbor's gene expression and the gene expression of the gene, mimicking some biophysics. Also, we allow there to be time-based autocorrelation to build up transcript. And we can also build it up over a period of time to support hysteresis. So this attention network, effectively, if you read it out, gives us a time-varying graph that supports biophysical effects like lags between gene interaction and buildup of transcripts. This again gives us a time instantaneous derivative that we use in an ODE solver. And we can further penalize the attention graph that it learns because we can say the graph has to be close to a prior graph and that it has to be sparse. So using this, we can actually get a readout of a network. And actually, this is kind of mechanistic. It's modeling at the level of the gene. So we can actually put a perturbation into the gene, and you can watch this perturbation kind of cascade. And that should give you some idea of how it'll affect downstream genes. So we're trying to do this, for example, in our embryoid body data set. And we can see how if you perturb one gene A, gene B changes, and then sometimes it comes back up because there's another regulating partner. And it seems to us that as we get more and more perturbation data, we can create a feedback loop where maybe we could use this to actually retrain Routini, not only to predict the trajectories, but also to predict perturbations. Currently, we haven't come across data sets that are both sort of time varying and have a lot of perturbations. Again, we've tested this on simulations, Sergio, as well as dynamic system simulations. And we've compared our networks to other networks, but it should be mentioned that all these other networks types are static. So we kind of compared an average of our networks to these other networks. Ongoing work in both MioFlow and Routini is to make them spatially aware, because now we have spatial transcriptomics that tells us what's around a cell. So we can incorporate spatial information and we can see how cellular trajectories change based on the microenvironment. So this is a key ongoing work that I have. So if you're interested in this, so you can let me know. This has also been used to figure out functional connectivity in neuronal networks. So we used something called a nest simulator. It's a network of coupled oscillators, basically, is the simulation model. And we can actually in silico perturb these and see what happens. In the real life, you can maybe train it using optogenetic perturbations of a mouse or something that you're measuring this in. And we also have results for that. So now that we have these dynamics, and I know I lost some time, we have a question of how do we characterize these dynamics? So we already saw in the cellular case, the different dynamics correspond to different trajectories. So some may be horrible, aggressive cancer, and some may be just on their way to dying or something like that. But it's a little more complicated when you apply this kind of technique to neuroscience data, because there's many, you guys have all heard of neurodiversity. There's many different kinds of neuronal regimes that are happening. So in order to make this amenable for learning, so in order to understand what the dynamics are in a neuronal data set, we sought to come up with a way of detecting dynamic patterns as well as quantifying them. And these patterns can be inferred by something like routine, or generated, or actually measured. So in order to do that, we use graph-based learning in a little bit of a different way. We modeled the brain itself as a graph. So in the cellular case, we modeled the gene regulation as a graph. Now we're modeling the brain as a graph. The brain activity is considered to be a signal on the graph. And we want intelligent ways of representing signals on these graphs, which take on very complex patterns. So the first idea would be to use something like a graph neural network. It turns out that they are not good at capturing global signaling properties along these. He's laughing. Did I misspell now? Okay. So again, apologies to Friedrich, but he also helped with this work. So we created kind of an alternative to graph neural networks that we've been working on over many, many years, with the latest iteration of this being something called BlissNet that was published earlier this year in AI Stats, and learnable geometric scattering, which I'm going to cover, whose latest version was published earlier this year in Transactions on Signal Processing. So the idea here is a lot of biological data can be modeled as a graph, and you can try using geometric deep learning. These are basically GNNs, and they in some sense generalize operations that were successful on images like convolution and pooling. But it turns out sort of simpler GNNs have these operations that you discretely say. They aggregate messages and update them. So basically each node in the graph takes messages from its neighboring node, and then it can create a nonlinear transformation that updates the messages on each node. And it turns out that just alternating between these using local message passing like this has some problems. And there's a lot of problems that you might have heard of: oversmoothing, underreaching, squashing, etc. But in our case, it also has another problem, that it's not super expressive of the global patterns of signals on genes. So one problem that we might run into is that it's not very sensitive to the underlying structure. Connectivity. These two connectivity graphs, then you can see that, for example, oh, I lost the, from a particular point, local message passing would just locally aggregate messages, and locally most of these nodes are similar. But if we use graph diffusion, which is an idea that I talked to you about before, we can see that this here diffuses differently on this graph than this graph. So it becomes very sensitive to the underlying structure of the graph. So this idea of using different scale diffusion, one step versus 2 steps. It's stuck again. Is it moving in online? Yeah. What?
Just stop sharing and go directly to the graph diffusion.
Now it's saying didn't even come up with that screen. Oh, there it is.
Maybe it's because you have like many windows open that it's lagging.
Think so?
I don't know. Did you select entire screen?
I did.
Yeah. Okay.
I thought I did.
Ça bougeait pas là.
Ouais, mais là, ça va complètement sur la... En fait, non, ça bougeait là, ça bougeait en live, et ça bougeait ici.
Mais ça, c'est connecté là, c'est impossible. C'est bizarre.
Ok, alors je crois que c'est en live, parce que sur mon ordi en live, ça bougeait.
Ouais.
Sur les...
Can you do it again so I can just know what you're talking about? Because I've literally never seen that ever. So.
Okay, I stopped sharing.
Ouais,
Now it's
maintenant, il
like
veut juste
stuck,
plus...
I think.
Ok.
Minutes or something. Okay, so the concept that captures so as I mentioned, you can use different scales of diffusion to capture signal patterning on a graph in a way that's sensitive to the underlying graph structure. And to do that, we actually literally create these diffusion wavelets that are just differences in different scales of diffusion. So it turns out these diffusion wavelets, which do bandpass filtering. So you can imagine just diffusion as a type of low-pass filtering, and a restricted type of diffusion is, as you know, what graph neural networks use for message passing. So instead of that, if you increase the expressivity of this a little bit and you're looking at the differences between scales of diffusion, then this gives you a diffusion wavelet, and it gives you a more multi-scaled way of looking at signal aggregation on a graph. So basically, what you can see is if you have sort of a complex signal on a graph, if you just smooth it away as we're doing with current graph neural networks, then you might not be able to distinguish between either nodes or the graph structure. But if you use different bands of wavelet, it can decompose this signal into different sub-signals that can be useful for classifying and doing things from the graph. So that's the heart of this geometric scattering method, which I now won't fully go over. The basic idea of the geometric scattering method is you take your signal and you transform it by alternating. This is a wavelet. Each of these is a wavelet. Graph wavelet transformation, nonlinearity, graph wavelet transformation, nonlinearity. And this creates a deep neural network that is like a graph neural network, but it's doing this diffusion-based message passing, if you will. And so each of these creates sort of a scattering path, and at the end you have different signals that come out of each of the levels of these. And what you can do to get a whole graph-based representation is you can aggregate these signals over the graph, and you can use statistical moment aggregation. So we wanted to put this into the frame of an end-to-end learnable network. And this is the one that was in the transactions of signal processing. So we have a neural network that recurrently applies diffusion to get different scales of diffusion. And you collect them in the form of these wavelets whose scales we specifically learn. And then once we have these signals, we can take, we can aggregate all the signals using the moment aggregation. So that's learnable geometric scattering. So we've applied learnable geometric scattering for a wide variety of tasks, mostly biomedical, autoencoding to learn representations of complex graphs, generative models to generate graphs, classifiers. Recently, we've also been changing the nonlinearity from being a modulus to two ReLUs, a positive and a negative ReLU. So when we do that, we can prove it's more expressive and that it's very good at capturing differences between signals on graphs. So the modulus nonlinearity, for example, can't tell the difference between certain kinds of signals with positives and negatives. And you see this version, which is called BlissNet, is more distinguishing and more expressive for different signals, particularly neuroscientific signals on graphs. Currently, we're also looking at other types of dynamics other than the diffusion dynamic to change the dynamic message passing on the graph. And some of the ones that we're doing are heat dynamics, wave dynamics, as well as dynamics that can be made to be chaotic or on the edge of chaos. And this is all to get more richer, more complex representations of signals on graph. And this paper is up in preprint, also it's under review. And what we've shown are that this can recover the graph topology, among other things, with increased efficiency. So what we're using this kind of graph neural network framework for is to classify, and so it's to represent and classify dynamic signals in the brain. So here's an example. So we have calcium signaling in the brain where cells communicate with one another via calcium. And the calcium signals can kind of look like this, which looks kind of crazy or random or something like that. And these particular calcium signals are in your epithelial cells, which some people didn't know actually transmit calcium. So you might be wondering, is this random? You know, what's going on? Is there coordination? But what we can show is that we can represent these signals using geometric scattering, and then we can collect the scattering representation over time to create a trajectory. And then we can quantify that trajectory using topological data analysis. And these 4 steps are called geometric scattering trajectory homology. And this can give us a way of not only classifying the dynamics, but also coming up with models that can recreate the dynamics. So I'm going to skip over a bunch of these details. If you're not familiar with homology, a really simple way of capturing homology is persistence diagrams, where if you have a bunch of data, like our scattering representations collected in low-dimensional space, you can grow an epsilon ball around them and count the number of connected components, cycles, and other topological features that emerge from this process. And this is a multi-scale characterization. So what we did was we tried to compare different dynamic regimes. So we compared calcium signaling in epithelial cells to calcium signaling in neuronal cells. And you can see the trajectories look vastly different. And it seems like in the epithelial cells, the calcium is globally coordinated. It's creating a global sweeping trajectory that sometimes loops. And it turns out it has a very specific function. It's checking for homeostasis. So if any cell is injured or stalled, this sweep gets perturbed. But in the visual cortex, it's constantly interacting with the outside world, reacting to stimuli, so it gets this more chaotic trajectory, which we can easily pick out. To establish that this is indeed some kind of homeostatic mechanism, we tried to stall cells in certain phase of the cell cycle, and you immediately see that these sweeping calcium traces become more perturbed. And not only that, you can classify trajectories based on whether they're stalled or if they're smooth. And the most interesting kind of thing is we were then able to try to mimic some of these calcium trajectories by taking calcium trajectory-based mathematical models and using our GSTH representation to generate parameters of these mathematical models. So we can use it to reverse engineer these trajectories and simulate more trajectories like this. So overall, this last story that I told you was about what you do after you get the dynamics. You can classify them, you can quantify them, represent them, and also regenerate them. And the way we basically did all of this was to use another graph neural network that captures the patterns and then quantifies the trajectories, as well as then goes on to have a predictive head that predicts these kinds of parameters. So with that, I'll end my talk. So if you're interested in anything I was talking about today, check out our GitHub, our website. And of course, I want to thank all of my collaborators, including people's pictures that I haven't put here. All right, thank you.
We have a bit of time for questions, if there are some. Lina, do you have a mic?
Yes, yeah. Can you hear me? Hello? Hello?
Yeah, that's your question.
Can you hear me?
Yeah, we can hear you.
Thank you so much. Thank you for the great presentation. I wanted to ask about my MioFlow algorithm and how does it mitigate batch effects. For example, is it possible to trace the trajectories of the same tissue cell type across multiple single-cell RNA-seq data sets?
Yeah, so MioFlow by itself doesn't mitigate batch effects. So there's many batch normalization methods or sort of domain transfer methods that can translate from one batch to another. But it is possible to take cells from different sources and pool them to run MioFlow. Actually, a lot of our own experiments that we're currently running, some of which I've shown you, are generated from different mice and things like that. So the batch normalization would be sort of a pre-processing step that you would do. First off, you'd have to assess if there is batch effect. Sometimes there isn't, even if the sources are measured at different times and things like that.
Okay, thank you.
Mike here. Since you're interested in both neurophysiology and transcriptomics, amongst other genomic modalities, I was just curious if you knew or have worked with patch-seq data.
I've heard of it, but can you remind me what it is?
Yeah, it's pretty cool. So it's it starts with the technology for doing single-cell patch recordings for measuring electrophysiological activity from single cells. And the idea is that you're kind of protruding a patch into the cell, like a recording device. And with patch-seq, you end up.
So, but then you kill the cell to sequence it or something?
Well, yeah. Then at the end, you absorb the nucleus through the patch that was originally used for.
No, this sounds fascinating.
Yeah,
if there's data sets out there, I'd love to hear about it. So.
Yeah, I think there are a few like open data sets, but there are a lot of kind of looking into that. So you get the transcriptomic profiles of these cells alongside I don't know what's
activity measurements or something.
Yeah.
Neurophysiological recordings are usually very time resolved, so that would be cool. But I'd have to adjust the time of the
time scales are completely different.
Totally different.
So then it's like a time scale within another time scale.
Sure, yeah, but that can be handled. Actually, we have some work looking at that just between two different neurophysiological modalities. So we've been trying to translate EEG to fMRI, and they're also like that. EEG has a lot more spatial temporal resolution. fMRI has more spatial resolution.
Yeah, I've worked on that too. EEG to fMRI. So it's hard.
It is.
All right, thank you.
Any other question? Let's see.
Thank you. That was a very interesting talk. I have a rather convoluted question, and I'm not entirely sure how to formulate it. But you're probably familiar with things like Waddington landscape theory, like more like abstract views of cell trajectories. And I was wondering, when you derive a network, when you infer a network, say, it seems to me, or I don't know actually. It seems you're going to get some kind of relatively local trajectories in,
relatively
local trajectories.
Trajectories.
Yes, in the sense that you're going to follow a pathway, like a differentiation pathway. But if you think about things like landscape or Waddington landscape, this is really capturing more like longer scale, like more.
I would argue the whole reason we made MioFlow is to have longer range trajectories rather than local ones like RNA velocity. That's why I was showing you the difference in vast amounts of time.
But what I mean by it's not only trajectories; it's really about the landscape. You're considering the manifold itself. Like how, I don't get a good sense of how you could describe, say, something like a landscape.
So we're not trying to explicitly model the energy landscape, because among other things, so the people who I've seen that try to model the energy landscape also have this assumptions that cells always go down the energy landscape. But there's no reason for that. Cells eat food, so they can go up, down, in, out, anywhere they want. So I haven't been trying to model the energy landscape. I'm just trying to model the cellular pathways. But I do model a manifold of viable states, but these trajectories are not local in them. No, there's no reason for them to be. I'm measuring them over vast periods of time where they're changing a lot, sometimes entirely. There is a question on there. I saw it.
So Surya is asking if you could explain a bit more why grandeur causality fails and why we need the Ritini network.
I wouldn't say grandeur causality necessarily fails, but it's a fairly simple kind of statistical measure that we were using to derive a static network. And the idea of Ritini was really to get a dynamic network. So Ritini gives you time-varying network by way of the time-varying attention mechanisms. And also, it's kind of based on our prior and initialization. It's the network that's kind of closest to our prior, but also explains the trajectory. So it's not necessarily kind of a probabilistic notion so much as this gene regulatory network is sort of the best explanation we have for the way the dynamics are proceeding. So I thought that had less assumptions.
And asking if...
But in terms of pure practical output, Ritini gives you a different network every time point.
Okay. The answer is if dynamic network is the only target, why not use a deep Kalman filter?
So we wanted to train the network to predict the trajectory. So I don't know, maybe it's possible to do it with that, but we didn't see a way. So we wanted to train the network that predicts our continuous trajectories. So that's the main prediction task of Ritini. It's bootstrapped to our trajectory inference.
All right. Any other question? All right, so let's thank Smita again. And sorry for the slide sharing.
Oh, sorry about that too. It must have been annoying for people to see. So, I don't know if it's the fault of the tabs, maybe. I like the infinite mirrors.